A very good afternoon to everyone. Myself, Dr. Balamurali Mohan, Assistant Professor, Department of Pharmacy Practice, LAXMAN Reddy Institute of Pharmacy, and coordinator for this Pharmaconverse webinar series. On behalf of our college and the organizing committee, it is my privilege to welcome you all for today's webinar. This is our 6th webinar. With a tremendous response and support from all the participants across the country, we have successfully completed 5 webinars on various topics delivered by eminent speakers from different fields. So without any delay, I would like to start today's session by requesting our respected principal, Dr. K.S. Murali Krishna sir, to address the participants.
Good afternoon, all the participants, and good afternoon, today's guest speaker, Dr. Amit Asthana ji. This is our 6th session of our webinar series, Pharmaconverse. Really, I'm very happy to have such an expert who is the principal scientist at CSIR-CCMB, Hyderabad. Amidst tight schedule at his organization, he has accepted our request to be a guest speaker for this session. And I hope all the participants will have a wonderful takeaway by his talk. Now I ask Balamurali to continue the session further.
Yes, sir. Thank you so much. So for today's webinar, as our principal sir said, we have Dr. Amit Asthana, who is working as a principal scientist in Center for Cellular and Molecular Biology. Now I request Dr. Nalini Kanta Sahu, Professor, HOD, and our webinar series organizing secretary to introduce guest speaker. Thank you.
Thank you, Dr. Balamurali, for giving this opportunity. I'm really happy for this reading out the biodata of our esteemed guest today. So very good afternoon, sir. Very good afternoon. And very good afternoon to all the participants. And good afternoon to our respected principal, sir, who is always behind us and encouraging to invite such eminent persons all over world known him. So Dr. Amit Asthana sir, I think this is a known face, and his name also known to all the people because he is an achiever of so many patents, and he has done 4 postdoctoral studies. So really happy sir to have you on this platform today. So currently Dr. Amit Asthana sir is working as principal scientist at Center for Cellular and Molecular Biology, CSIR-CCMB, Hyderabad. He obtained his PhD in chemistry from Barkatullah University, and he has done his postdoc in 4 different countries. And he has more than 20 years of postdoctoral experience in micro and nanotechnology, microfluidic devices designing, fabrications, characterizations, hot embossing, clean room operations, and paper microfluidic. And recently he has published a paper on microfluidics. He has done his postdoctoral studies in 4 different reputed university like Netherlands Institute Curie and University of Illinois and your Chungam National University, South Korea, University of Queensland, Australia. And he is the fellow of Telangana Academy of Science. He is recipient of many fellowships, including Senior Research Fellowship, CSIR, Curie Fellowship, France, Brainpool Fellowship, Korea, and Ramalingam Swami Return Fellowship, India. And Dr. Asthana has several publications in journals of international repute and international conferences. His research interests include biomaterials, drug delivery, applications of micro and nano fabrications, micro and nanofluidics, digital microfluidics, and a wide range of microfluidic tools for biological studies. Dr. Asthana has been instrumental in establishing PAHL Bio-Nanotherapeutics Private Limited, a startup established with the funding of BIRAC under Bio Initiative Grant at IKP Park, Hyderabad. Dr. Asthana had been associated with other biomedical devices startups as scientific advisors, which includes Axio Biosciences, Gujarat, and its inception with Theranosys Life Sciences, Hyderabad. So with this short and brief introduction, I like to invite our guest speaker today, to take over the session and continue his lecture on fabrication of your paper-based device and its biomedical applications. Please, sir.
Thank you, Nalini, for such a nice introduction. And I would like to thank Professor Murli Krishna, Principal of MLR College, for inviting me for this web series, 6 web series, and to Nalini and also to Professor Dr. Murli Mohan. Thanks a lot. So if it's okay, I will start. I will share my slide and we can start the talk right away.
Yes, sir.
Yeah.
Thanks a lot. So I hope you can see the slides now.
Yes, sir. We can see. You can start your presentation.
So as mentioned to all of you, the topic of my talk is paper-based microfluidic device and their application. And if you have any questions, we can have a small question answer session also. But if you have any question, you can always write to me on my email address, which is given below here, aa@ccmb.res.in. So what I've done is I have divided my talk in 3 major parts. What is the problem currently? So affordability of healthcare, that especially in diagnostic, is what I'm going to address in few slides. Then I will give you a solution, which many of you probably know, paper-based microfluidic devices. So we will talk about why paper, how we can use them, what we can do, and how we can fabricate them. So I hope eventually after end of this talk, you will be able to make your own microfluidic devices, paper-based microfluidic devices. And then I will show you some of the application like blood typing, total lipid profiling, early detection of pregnancy in cattle, surface enhanced Raman microscopy substrate, which we have recently developed, and another project which is on viscosity measurement. If time permits, we'll go to that. So now in healthcare is it's our basic need, and at the moment POCs are in demand. So in India we have achieved tremendous success in the area of healthcare, but it is sort of a patchy one. In resource limited area like villages and small cities, they will not have good diagnostic tools. So we need something which is reachable to mass, which is reachable to area where modern healthcare methodology is not there. And that is what probably we will be trying. So current in vitro market is quite big, as you can see here. I will not repeat the numbers, and it is growing very rapidly. And from 4.75 now, I think it is 6 point something, and it will reach to larger amount. And in India, we are reaching to a population which is susceptible to various diseases. Our population is gradually getting older, though we are the youngest workforce at the moment, but still we are reaching there. And sorry, the regular diagnostic tests are more or less driven by regular diagnostic tests, which you have to go and get it done. Whether you are suffering from a disease or you want to know how you are, how fit you are, you have to go for tests. And if you can do those tests in home, nothing like that. Current in vitro market, the largest shareholder in IVD market is point of care devices, and those point of care devices are mainly pregnancy kit, ICV detection kit, or similar dengue detection kit. They are, if you probably have seen cassette kind of thing where you put a drop of sample and then you see the color change. Those are all point of care devices and they are called lateral flow devices. We'll discuss about that in detail later on. I'll just skip this and I'll come to the next slide. The conventional analytical tools which are shown here, like PCR and electrochemical detection system and other small influenza test kits and glucometer, Glucometer is the cheapest one and which is the closest one for POC, low cost POC devices. But most of them require instrument and they are not that affordable. What we are targeting is something which is mentioned by WHO in their guideline. It is called assured devices, which is affordable, sensitive, specific, user friendly, rapid, and robust. Equipment free is more important, and delivery to end users. That means that device should reach to end user. You can go to a medical shop and ask for a device, take the device and do the test by yourself by taking urine sample, saliva, or blood sample, whatever you want, even tear. So microfluidics is one way of doing it. It can bring down the cost and dimension. Some of these devices which are shown here, like the glucometer, is very close to microfluidic devices. If not microfluidic devices, they are very close to microfluidic devices, but a lot has to be done. And these kind of devices made by microfluidics can easily be utilized in low resource community. Now, microfluidic devices can be made on different substrates. It started with silicon, then people started working on glass, then plastic, then textile. If any of you know Achiria Labs, they are developing diagnostic kits on textile or cloth, and we and some other companies in India and abroad are trying to work on paper-based devices. The initial one was from George Whiteside's group, and the company was called as Diagnostic for All. They are now closed down, and why they are closed down, we will discuss that. But there are some good companies coming up in India. One of them is in Chennai, called as no, actually in Odisha, in Bhubaneswar, and also they have some part in Chennai called as Micro Life Sciences. So they are manufacturing paper-based devices, and they are into market now. They will be in market very soon. Apart from that, there is Pragati printers. They are helping us, and we are helping them to increase the visibility of these devices in market. Mass production of these devices is very hard, and we will be taking their help in future because they are expert in printing. So a POC device typically is divided into 4 parts, but our POC device is only these 2 parts. So you have a sample, you have to recognize a sample. In sample, for recognition, you can have enzyme, antibodies, microorganisms, cells, nucleic acid, aptamers. Nucleic and aptamers are similar, nanoparticles. Then you have electrodes, transistors. These are for single transducers, and single processing is done with filters and multiplexes and AD converters, etc., which are part of glucometer also. But we are not considering these part. If we include this, that means it is not equipment free. What we are trying to do is we are making a device where you can do recognition and immediately read it out, and mostly it will be a naked eye detection, but you can also use cell based detection image analysis system. So current all the devices are using nitrocellulose membranes. So nitrocellulose membrane is quite near to what we are trying to do. Recognition area where you are testing everything, or test zone, whatever you want to call it, and the readout, which is the color change. Okay. Now these nitrocellulose membrane, plastic, glass, silicon, they are all expensive. They are not that affordable, and still these devices, except lateral flow devices, are not off the shelf available. So we moved to paper microfluidic devices. We thought paper could be a very good substrate, and there are already a lot of literature available. Why we want to use paper? First of all, paper is available everywhere. We are using paper for all sorts of things. So you can even make paper devices on toilet paper, kitchen towels, anything you can make a paper device out of. So they are very inexpensive. They are not that expensive paper if you want to take, and they are biodegradable because they are made up of cellulose, or you can burn them away. And they are portable, so you can make very small devices, which I will show you in some time. And sample movement, you don't require any pumping system or an instrument. Basically, you put a drop, and then solution will move automatically towards the detection zone where you want to detect everything, or reaction zone, because of the capillary reaction. There are these paper are made of fibers. These fibers are entangled to each other, and the gap between them create a capillary. So you put a solution, and because of pressure difference between two sides, solution will move in the capillary and it will reach to the end of the capillary. We'll see one video also to clarify this. So paper-based devices, first portion which you should know how you can fabricate these devices. We have wax printer in the lab, XY plotter we can use, and laser printer, and there are various other methods which n number of people are using, n number of researchers are using, n number of methods are there. Like Pragati printer is using completely different method, and which is more suitable for bulk production. And what we are doing is something which can be a lab prototype kind of thing. So if you want to make a device, I'm pretty sure that after this talk you will be able to make a paper-based microfluidic device in your lab. Detection strategy, we are using Raman microscopy electrochemical detection system, but we are concentrating only on naked eye detection. That is what should go into market. If you have a detection system, that means you don't have an equipment-free system with you. So for paper device, cellulose is relatively inert material, very less reactivity. So you have to bioactivate the paper for immobilization of different molecules which you want to use, like enzyme, aptamers, nanobodies. Nanobodies are nothing but antibodies, smaller one, half shape. And these nanobodies are actually camel antibodies mostly, and other reagents, whatever you want to use. So idea is, if you put a drop, I don't know how many of you have used ink pen. When we were kid, we used to have fountain pens, and if during that time paper technology was not that advanced, so paper if you put a drop of ink on your paper, it used to spread everywhere. Right, like it's shown here. This is a drop of ink which is spread everywhere on a paper towel, and it's going anywhere. Now, if somehow you can make hydrophilic and hydrophobic barriers, then you immobilize your enzyme, antibodies, whatever you want to use as a recognition molecule, or at the end, when you put the sample will move within these boundaries. That is what we are targeting. How we will make this and how it works, we will show in coming few slides. So how you can make microfluidic devices? We have a wax printer. We are using wax printer. In IIT Hyderabad, IIT Chennai, and IIT Kharagpur, they are using another printer. It's a laser printer, HP laser printer. I don't remember the model number, but that also have wax. Now in this particular system, there is a wax cube. Unfortunately, I don't have a picture of those wax cubes here, but there is a wax cube. There are 4 wax cubes which sit somewhere there inside the machine, and the roller is heated. That wax fall on the roller, and then you can just make. You can have a printout like a usual printout, but when you heat it, it spreads a bit. Now your wax actually is falling on the paper on the top. But if you want to make a complete device, what you have to do is that this wax should go all the way through to the other side of the paper. If it is only on the top, then because of the capillary action, your devices will leak. Right here, there is one device. If I put solution here, if it is not heated, if the wax is not percolated to the other side of the paper, then you will see leakage on these parts. Right. So what we do is we take the printout. After taking the printout, we heat it at around 120 degrees for 2 minutes, just for 2 minutes. Then this wax melts and percolate down to the end of the other side of the paper, and also it weaks on both the directions, on the horizontal as well as on vertical. So when we design in such a way that if you have to make a 2 mm line, 2 mm channel, then we make a 3 mm design, because it will weak that much. So any method, if you make the devices with the HP printer or with the wax printer or with XY plotter using a permanent ink pen, which I will show you, you have to use that. So we make the design first, we take the printout, then we heat this print at 100 degrees, 120 degrees for 2 minutes. This is our paper device. Then you can put sample, working reagent, use a plotter, sorry, a scanner or a camera to measure, and you can quantify also. Okay, this is the usual way. Or if you have a laser cutter like this one, which is also not very expensive, you can cut various designs, glue them together, and you have a device ready. This is the device which we have used for blood typing. The easiest one is XY plotter. XY plotters are, this one is an expensive one, but there are very affordable XY plotter available. What we do is we make the design, we put a pen there. I will show you the pen on other slide. That pen can be any permanent ink pen or the ink pen, like we have developed our own ink. We use that ink. You write with that ink. This ink is organic, the resin in the ink is dissolved in organic solvent. So as soon as you write on the paper, the ink will percolate, the resin will percolate down towards the end of the paper and also in the surrounding area. So you don't have to heat it. That means if you are not heating it at 120 degrees, your integrity of your cellulose paper is intact. You don't have to worry about your paper getting burned, charred, or getting changing colors. So this is one advantage, and this is the single step method. If you don't have an XY plotter, you can just use your hand and draw something and test your idea. So this is the front side, this is the back side, both look similar, and you can do the same thing: sample, working, reagent, scanning, image analysis to quantify your analyte. If you don't want to quantify, just by seeing the color change, you can identify whether it is high, low, or yes or no. Right. So this is the plotter which we are using, and we are using Cellular pen. So this is a secret which I'm sharing with all of you. Don't use any permanent ink pen, but try to get hold of Leuco color Cellular pen, permanent ink pens, and this kind of holder is available with these printer and other printers also. You can put this pen inside the holder, and then you can make devices like we have shown here. Now it is very important when you are doing your work to know what exactly you want to do. If you want to analyze some material, you need to know whether barrier efficiency of your devices are good or not. So we have done some barrier efficiency tests with glycerol and different ionic strength solution. Apart from that, we were targeting artificial tear, sweat, saliva, and blood. So we have tested this with our permanent ink pen devices. So you can see here on the right side, these devices don't leak at all, neither with artificial tears, nor with sweat, nor with saliva, also not with blood. Now we have tested these with organic solvent also, with methanol 70%. So these devices can hold, the one which I am showing here can hold up to 2 microliters of sample, only 2 microliters of sample. But you can see we are increasing, we are going double, we are going 4 times, we are going further. It is not leaking at all. On the other hand, if you take wax devices, they will leak. Right? So these devices work very nicely with methanol 70%. If you go to 100%, they will start leaking. This is ethanol. Ethanol is below, sorry. So this is up to twice the volume of what it can hold. These devices will not leak. After you increase further, you will see some amount of leak taking place in wax, because wax will start dissolving in organic solvent. While in case of methanol, it will start leaking. I mean, if you are using just 2 microliters, you are fine. Just 2.5, you are fine. But if you have more, then these devices will leak. So you have to remember what are your reagents and what is your sample, whether your sample is dissolved in aqueous solution or in non-aqueous solution, and based on that you should test your devices before you start doing your analysis, right? So this is with SDS. Obviously any detergent will dissolve resins and waxy material or lipid material, so it leaks. With NaCl, that means high ionic strength material, it will not leak at all. And if you increase the viscosity also, they will not leak. So these are some example I'm giving you, so you can test in various forms. You can make different structures and see if your devices are leaking or not. We have tested we have done these leak tests with various material, SDS, print detergent, glycerol, NaCl to see the whether ionic strength have any role, methanol, ethanol, isopropyl alcohol, acetone, with different grade of paper. We usually use Whatman paper grade, but many people use Whatman paper. Maximum people use Whatman paper grade 1, and different other papers also we have tested, and these are the analysis. And with Stadler pen, it works very nicely. It gives us very good result. Okay, so now we will move to a video. So you can see this is the empty device, and now we are filling this empty device with 100% methanol, and you will see it will not leak. So I'm putting this methanol on the paper, and you can see the solution moving in the direction where we wanted them to go. So this is our reservoir from where, or the input area, and somewhere there we can have detection. I will run this once more so that you can see. So I'm putting the sample on input area, and it should reach to the detection area there. And mind it, this is 100% methanol, sorry. Right, let's go further. So the first thing which we did, and actually we were able to publish it very recently. This is the so there should be a comma here. This is published just recently, a month or so in 2020, and this is a blood typing device. So we make the device, then we take a wax print. So you can see how sharp is the print. When we heat it, the size, if you see here and here, size is different now. Size will reduce because wax will percolate not only downwards but onto the other sides also. So that is why size decreases. And this particular device is bioactivated with various material. For this particular case, we have used chitosan, and once chitosan is we have treated this area with chitosan, then we immobilize antibodies. So this is A, B. Conventionally, A antibodies come as a blue color solution, and B comes as a yellow color solution, and RH will always be white in color, just for your information. So how we do this work is that we take blood samples, we collect blood samples from a dispensary, and then we put a drop on this antibody immobilized device where we have A, B, RH and control. We put a drop. Once this drop is spread to all the arms of the flower shaped device, then we wait for 2 minutes. After 2 minutes, we wash this device with saline, and we keep a tissue paper or a blotting paper underneath. You can see this tissue paper or blotting paper underneath. So whatever saline is there, it will be sucked away by the paper underneath. So if agglutination is taking place, then your capillaries of your paper will be closed. And they are closed, the RBCs will not move further in the paper, or they will not go downwards. They will just remain there. That will give us answer. So if there is agglutination reaction taking place, this area will remain red in the color because of RBC, and rest of the area will be washed. So this is the example of A positive sample. So you can see in C and B, it is washed away, but in A and RH, they are still there. So this is how we can test blood, and such devices will be very useful for emergency uses, and also for low settings. So you don't have to, usually you put antibodies, you put blood drop, you mix them, and if at all you use the same mixing utensil or a tissue or whatever capillary, then you are gone, because there will be cross contamination. There is no chance of cross contamination here, and the amount of antibodies which you are taking is 1/4 of what you usually take, or actually even less. So it's a very unique way, and the devices are not very expensive. Further, these antibodies, blood typing antibodies, are not stable enough at room temperature. But they can work for, if you keep them in refrigerator, you can use them for very long time. But if you keep them at room temperature, after 10 or 12 hours, they will not work. They will be denatured. So if you make these devices and they are not stable for 6-7 months, it is not useful. So what we did different immobilization process, and we have tried chitosan dry, chitosan with glutaraldehyde, chitosan with STTPP, which is a phosphate, triphosphate, NaOH. We have tried all these methods, and you can see AB positive and O negative results shown here. Right? Among them, we found that when we do immobilization on chitosan, which is cross-linked with NaOH, works better, and we get results up to 110 days. So our devices, what we make, are stable for 110 days at room temperature. If we keep them in refrigerator, they will work forever. Right? So these are some image analysis which we have done. So you can see they are very good up to 90 days, but they are good enough till 110 days also. So these are the results which we are showing here, and this is, you can read this paper for further details if you wish. Apart from that, we have developed a total lipid profile device also, where we can do all the lipid profiling in a single device. So this is the design flower-like device. You can have HDL, VDL, total lipid, control, and other tests done in a single shot. Again, this paper is available online. It was published in Sensor and Actuator B. You can do image analysis if you want to quantify by putting the device in scanner or taking the picture by camera. So these are some of our results, and you can see that for total cholesterol, whatever sample we got about correlation of 9.18. For LDL, correlation is even better. Though this is a very small sample number, but results are very impressive. So you can do all total cholesterol, HDL, LDL, TG, everything can be done with this method, and the results are very impressive. So this is another possible commercial product which can come into market, and there is a company which sells chitosan, catalytic, and their chitosan is very good compared to whatever we get in the market. So we are planning to take this further with them. We are just discussing with them. This is the work which I am very proud of, early detection of pregnancy in cattle. So one of my colleagues in my lab, Dr. Umapati, was working with big cats, and he was able to find a metabolite in the fecal sample, in the dung sample of big cats. Then he realized that if this is available in big cats, this particular analyte should be available also in domestic animal. And he did some work. He found that even in cow dung we can get this sample, but with different concentration. And he approached me, can we make a paper device? And we have made the paper device. We are soon planning to transfer this technology to someone who can make the devices, or we'll start our own company. So currently we have about this is the old Sensex. New Sensex is still not available. So based on this old Sensex, we have about 190 million cows, 13% of the world, and we have about 56% of buffaloes in India, which is 108 million buffaloes at that time. This count might have increased. The dairy industry turnover is quite high, as written here, and about 70 million farmers are actively involved in such kind of work. Traditionally, when you are doing pregnancy tests, you have to wait for 3 to 4 months, and how it is done is very interesting. You have to call a veterinarian. Veterinarian will put his hand inside, like shown here, inside the animal, and he has to feel the fetus. So if you have a very trained veterinarian, then he can tell you in 1.5 months. But that's also not enough. Or in Western countries, they can use ultrasound. Use of ultrasound in India itself is very limited. So these two things are quite late, and this farmers lose a lot of money, and they want to save the time. They want to shorten the intercalving period, and they can plan the artificial insemination if we can tell them early enough. So this potential marker which we have detected can detect pregnancy in cattle in 3 to 4 months. So we are saving a lot of time and money for these farmers. So how this device work? These are the initial steps. What we have done, conventional ELISA, when we do the same work, it takes about almost 15 hours. But when we do on paper, it takes only 15 to 20, 25 minutes. Now we have reduced it down further. So 15 hours to 30, more less than 30 minutes, 1 ml to just 60 microliter of complete sample as well as reagent. Idea is you collect the dung sample, extract the dung from the proprietary extraction solution which we have developed, put a drop on your paper. If you are a pregnant sample, then you will get only one color, but if you have a non-pregnant sample, you will get two lights. How and why, I will show you. So it is based on competitive ELISA. So on a substrate, on a paper substrate, we first immobilize the antigen, what we want to detect, the metabolite which we want to detect. We take the standard of that, we immobilize that on the paper device. Then we bring the antibodies which can detect this particular analyte, which is tagged with HRP. Now, if you bring the substrate, it will show you a color. In our case, we are using TMB, so TMB will give you blue color, right? This is a usual case, which will happen in the control. In the control, we will have standard antigens which are immobilized, and then you bring antibody with HRP, bring the substrate, and you see the color. Now, in real sample, the test sample, when you will put the sample there, in the solution, there will be already some antigen available from dung sample. These will react faster than what is immobilized on the paper. So these devices, these antibodies will react with antigen which is available in the sample, and when you will watch them, they will go away. Now, if you will bring substrate, then you will see less color compared to this one. Okay? Yeah, here color will be more, here color will be less, and this is what is happening here. So this is how our devices work. So if you have a non-pregnant sample, you will see color in both of them. If you have a pregnant sample, you will see color only in one, and another one will be either very light or there will be no color. So these are some of our old results. You can see less color. That means this is a pregnant sample. There are color in both the control and T. That means a pregnant sample. Though it was really hard to differentiate it initially, now we have better devices wherein you can see non-pregnant sample is showing you very dark blue color, while a pregnant sample is showing in T very less color or no color. So this is how our devices work. Our current device which we are using is a origami kind of design, where you don't have to put sample one by one, but you can have just you have to just put sample will move from this channel to this one, from this channel to this particular zone. Then from here it will move to a zone where we are immobilizing antibody. Okay, and once antibody is immobilized, then we will put buffer solution. Buffer solution will fall here somewhere, and it will move in all three arms of the microfluidic device, and it will come to first, actually it should come here to wash the antibodies which are not immobilized, stored there, and from there it will go to the detection part where you will have detection. So once you detect it, you will get answer like this. So you're putting sample, you are putting the buffer there. If you are a pregnant sample, then there will be less color compared to control, and if you have a non-pregnant sample, you have color in both the paper devices. So these devices are, if we sell them for 100 rupees, the total market is about 450 crore rupees, a big market. I don't want to take you to this all these things. We'll quickly move to next idea. As I mentioned, another work which we are doing is surface enhanced Raman microscopy. So this is something very well known now. Raman microscopy is a fingerprint technology for any molecule, but it is very less sensitive. You need a lot of sample to get the fingerprint of a particular molecule. But if somehow you can enhance the Raman scattering, then you can get the answer pretty easily. So I will not go into detail, but I will show you something which will give you much better idea. So you have a sample, and if you put this sample or molecule in a laser light, you will get Raman scattering. So you can see the peaks are very small. So if you know the fingerprints, you can calculate the scattering, shift in the scattering, and you can identify a molecule. Now, this is very weak signal, difficult to detect. Now, if you have a nanoparticle and you put a if you put a light on nanoparticle, this nanoparticle will generate an electrochemical field, and this electrochemical field is called as plasmon. This plasmon will have lot more energy compared to the laser falling on this. So that's why the molecule will start vibrating faster than when it is the Raman scattering there, I see this kind of work. So without nanoparticle, these are the are you able to hear? I think I just got a message that I was disconnected for a while. Nalini, hello.
Yes, sir. Now it is okay.
So should I repeat it or?
Yes, just from previous slide.
So this one?
Yeah, previous one, sir. No.
Okay. So these are the paper devices which we have made. So as I showed you in early slide, these are ink to make them hydrophobic, and this area where we are developing the nanoparticle. So you can see gold nanoparticles are getting prepared here. Once they are dried, these are the actual devices which we will use for the analysis. When we take the SEM picture, these are the Milky Way kind of picture where you can see gold nanoparticle deposited on fiber. This is another picture where gold nanoparticles are deposited on a paper fiber. When we see under the microscope, they look like this. When we do Raman scattering measurement, you can see that we are getting red color Raman intensity everywhere. The black area where you don't see intensity are not focused. If you focus there, you will see intensity there also. In among this area, we will choose this kind of area where light intensity is very high, or the intensity from the molecule is very high. This one, that one. So there are several places. If I analyze them, I will get my signal. So this is the signal of R6G, rhodamine 6G, as a molecule of interest we have used, and we found that compared to normal paper, we get far better intensity, about 1,000 times more intensity compared to that. We can go down with this method to 1 picomolar level, and if we prepare even closed nanoparticle with each other, then we can go femtomolar also. When we were doing this work, we called some vendors to check their handheld Raman microscopy if we can use our devices with them. So one of them was BW Tech, which is now under Metro Home. Metro Homes is selling this particular instrument. And when we tested, they were also using some paper devices. I think those were silver nanoparticle devices. Their result was here, and this is what we got with our device. So it is about 300 times more sensitive than the paper device currently used in the market. And this is all patented. This we have patented recently in PCT. It is published, and also Indian patent is also published. Now, another possibility when we were doing this work, we found that we can use these devices for probably viscosity measurement. You all know how we can do viscosity measurement. So if you have a test molecule, you can calculate the viscosity of that test molecule against this viscosity of water, the time taken by the solution to travel from this area to this area in a conventional viscosity measurement experiment. The density of that material and time taken by that material to move from here to there, and then you divide it by water density and water time, how much time water has taken to travel, then you can easily calculate the viscosity. So we did the same thing. We made a device like this shown here. Again, this area is marked with permanent ink pen there also, so that it will not leak. These are arms to support the paper devices. If I put a solution here, and then when it will start moving in this channel, we measure the time taken by that material, that solution to reach from this point to that point, and that is what give us viscosity. Let's see if it works for us. This is the complete setup which we are using. So as soon as we put solution here, we start the stopwatch and we measure the temperature, at what temperature it is we are doing the experiment. So this is a complete setup, and if you want to go through the detail, you can see this paper in Analytical Chemica Acta in 2018, where we have published this. So this is my device, and I have tested this device with different material, compared it with Oswald viscosity meter, which I have shown you in early slide. We have done 10 repetition for glycerol. We have used glycerol from 0% to that means 0% means water, to 80% glycerol, and these are our result: average time, average viscosity, average time, average viscosity with Ostwald viscosity meter. And when we plot them to see the correlation, we get highly impressive correlation between calculated Ostwald viscosity versus calculated viscosity with paper devices. It is 0.995. So then we thought we will try it with sucrose also. So we tried this with sucrose from 10% to 60%. These are results. Again, paper device. We have repeated this experiment 10 times with Ostwald viscosity meter 3 times, and these are our result. Again, very impressive. So we can use paper devices for viscosity measurement, and you can use these for saliva viscosity measurement, as well as for blood viscosity measurement, urine viscosity measurement. In labs, you can use them if you don't have a Ostwald viscosity meter, or you have broke your viscosity meter, then you can use these simple paper devices and you measure the viscosity very easily. So with that, I would like to end my talk here. So currently we are only 2 people in my lab since March. Dr. Ira Bhatnagar is a scientist, principal scientist also. She's also working with me, and myself, we are doing most of the work. And we are just, because of COVID, we have at the moment no projects. So we are soon going to start some work on COVID. Most of my funding is from Department of Biotechnology and CSIR, and also DSIR, our parental organization. This is CCMB campus. This is an old picture. This is how CCMB look like. It is divided into 3 parts. We have east, west, and south zone, where we have labs, and in the center we have all the instrumentation facility. I keep on trying to change this picture. This is the old picture of our building where we are sitting. These floors are all working now. They are all completed. We have CRTDH incubation center here, and Atal incubation center there, and our labs are on first floor. where my lab is also there. Main campus, we also have another campus which is called as CCMB Annex 1. It's called Laboratory for Conservation of Endangered Species. So my colleague Dr. Umapati, with whom I have developed this pregnancy kit, works here. It's a beautiful building. Architecturally, it is marvelous, and it won a lot of awards also. With this, I will end my talk, and if you have any question, I will be very happy to answer those questions.
Yes, thank you, sir. It was really a very wonderful presentation, and I am looking forward for the questions from the attendees. So attendees, if you have any questions, please post your questions in the question box. So our guest is ready to answer all of your questions. And yeah, sir, there are already a few questions. Can I read it for you?
Sure, please.
Yes, sir. So the first question is, what are the types of paper fabricators and how we use in drugs?
What are the types of
Paper fabricators
and
how we use in drugs?
I don't understand the question, but what I understand is there any fabricator for paper devices? So yeah, there is one in India, nowhere else there is anyone. It's called Pragati Printer. If you mail me, I can put you in contact with them. If you want to make the devices by yourself, you can make the devices by yourself using permanent ink pen, easiest way. You can just, if you don't have XY plotter, just use hand and draw. Go slowly. If you go slowly and make your design very slowly, the ink will percolate to the other side of the paper, and once it is percolated to the other side of the paper, you can easily use them. It will take 5 minutes to make a device. Okay, so you can use by yourself. If you want to commercially get it, a Pragati printer is possibility. You can send me an email aa@ccmb.res.in, and I can hook you up with them, and they can probably make devices based on what you want.
Yeah, okay, sir.
Next
Thank
question.
you. Then another question is, to what extent paper-based diagnosis is effective and how to increase the shelf life of antibodies?
Okay. So see, to best of my knowledge, there is not a single device. No, there is a device now in the field testing in India. Diagnostics all wanted to bring device, but they are closed now. They are the biggest player in this area, but they are not no more there, and their facilities are on sale in Boston if anybody wants to purchase it. There is a company started by young IITians from IIT Chennai. It's called Micro Life Sciences Limited, if I'm not wrong, and Abhishek Birla is the owner of the company and the co-founder of the company. You can search. He's winner of 2 Gandhian Awards. We are working together also. So I know that they are their devices are very effectively working, and they are working with a dairy industry, big dairy industry, and their devices are used for milk adulteration, and they are about 95% accurate. So they are only one which can come in the market at the moment. Even they are faster than us. Yeah, that's one question. Second question is antibody. It will depend on what material you're using. Like we have used chitosan, and we were able to show that it is up to 110 days they were. But recently we came across chitosan from a company, Canadian company called Ketolytic. They have a purest form of chitosan available with them. And importantly, they have chitosan which is 95 to 96% acetylated. So if you are taking that kind of chitosan, then I think you can increase it further. There are various other methods also for immobilization, but I can only tell you about chitosan, which works very nicely for us. If you want to immobilize aptamers, there are other methods, and stability is still people are trying, but 3 to 4 months they are easily stable on paper. With our devices, they are even more.
Okay,
maybe we can ask the next question.
Yes. Can this technique will be used for early detection of cancer?
In principle, yes. And there are some papers also, but a lot of work need to be done.
Okay. Can you please tell me the composition of paper?
Okay, this paper which we are using, cellulose paper, is mostly a pure form of cellulose. There is no other additives there, so it's a pure cellulose paper.
Okay, sir. Then another one is, to what extent paper-based fabricators useful for biomedical applications?
I didn't understand this question, Murli, frankly.
Okay.
So
Then,
at the moment, if you are asked, if he or she is asking whether there is any fabrication facility available to make paper devices with any kind of paper, so I told you already, you can make yourself. And if you want a company, Pragati Bioprinters is there, or Micro Life Sciences is there, or you can contact CCMB and we can help you in making the devices. Or if you have contact in IIT, IIT Madras or IIT Kharagpur, Professor Suman Chakrabarti or Chattopadhyay, I'm very bad with the name. His group is doing a lot of work. They are making paper-based fuel cells also, and also diagnostics.
okay, sir. Then another question is, paper-based microfluids are biodegradable, but they usually come in a plastic casing, which is not biodegradable on the commercial scale. So what other alternatives are there for the casing or cassette?
Well, there is not a single paper device available in the market. You are talking about nitrocellulose membrane. Yes, nitrocellulose membrane itself is, you can burn it out. Plastic, there is no alternate at the moment, but we are working. There is not much result at the moment, but paper itself can be used for the casing. So if this answers your question, for our pregnancy kit, we were trying to case it between lamination sheets. Lamination sheet, you can burn them very easily, so they are much easy to decompose or burned compared to plastic cassettes, where lateral flow devices come. So this is not lateral flow device at all. Please don't get confused. There are people even in literature, they mention if they work with nitrocellulose membrane, they will say I have worked on paper devices. No, not at all. These are not paper devices. They are nitrocellulose membrane. They have membrane. They have different structure. You don't have any capillaries. The solution move on the top of the membrane, not inside the paper device. So there is a vast difference. You people need to understand that nitrocellulose membrane is not paper device, not at all. Please.
Yes, sir. How chitosan increase the stability of antibody and prevent the denaturation at room temperature?
Yeah, fantastic question. Right. So what will happen? How antibodies will get decomposed or denatured? Why they are not active? So they have this kind of structure, right? And antibody. So this portion, if it fell down, it is closed, then you cannot recognize, right? You have to keep that open. Now, and also this portion should be up towards the analyte. So what we do is, when we immobilize chitosan, the orientation of these antibodies is theoretically on proper orientation, like this. Okay, so when you have chitosan holding your analyte like this, then the moisture will keep coming and falling on in between this area, right? So this area, this is all theoretical. I don't have any experimental proof for this, but for us it is working. So hopefully this, if you can prove, I am trying to prove also, if you can prove that this is the way it is working, this is how it is activated. So antibodies remain active because they have some water absorbed from the moisture, and that is why they are active. That is one probable reason. Apart from that, you can use some material which can block this area, but as soon as you put the sample, that will go away. That will increase the stability further. We are working on it, but that is another possibility. So chitosan, keep your antibodies on proper orientation. That is why they are more stable and they work much better than other methods.
Thank you, sir. Then, is there any application of this particular thing in present COVID situations?
Yes, there are groups working in Canada, US. I don't know in India how many people are working, but many people are trying with nitrocellulose membrane, but not a single device on paper in India at the moment. No one. I think some CSIR labs might have been working, but I'm not working. And there are some US universities, Canadian universities, especially Waterloo University, they are doing some work.
Okay, sir. Thank you. One last question. Which is the best wax printing option for printing pattern on different papers for microfluidic applications?
So I mentioned to you, wax printing is the best. We found that Stadler pen work very good. So you can use Stadler pen, or if you can purchase a HP printer. I don't remember the name of that printer because we have never used it, but that a particular model uses a toner which has a lot of wax. So that can also be utilized. And then Pragati printers have a completely different way of making devices. So they use hot embossing kind of method. So you can use them. There are several methods mentioned in the literature, but I will, if you are not familiar with these methods, use Stadler pen and XY plotter. Hope you people enjoyed this talk.
Yes, sir. With this, we have completed all the questions. Of course, few questions are repetitive, so I skipped those things. So with this, we have covered all the questions.
Okay.
So now I hand over to Dr. Sahu, sir, just to give a vote of thanks.
Thank you, Dr. S. Balamurali Mohan, again for giving me charge to give this important part of this thing. Actually, no duty is important than returning thanks, and especially today's speaker, Dr. Amit Asthana, sir, who has accepted our proposal and given a very nice and informative talk to all the participants. And I hope it will be very much useful for manufacturing this kind of devices with less cost, I hope. And in future also, the participants, they can take the help from Amit Asthana sir, and he is always helping kind of person in these things. So first of all, I like to thank our management for giving this facility, for encouragement, for conducting this webinar series. And as Dr. Balamurali said, that already you have completed our fifth series, and today is our sixth series. And it was a talk with eminent person like Amit Asthana sir. And next, I like to thank our respected principal sir, who is always encouraging us for doing all these hardships and to make all these kind of things successful, and to aware all these participants, all the teachers, all the people from the different domains to know all these kind of things, all these new kind of things. So thanks for that. And next is our today's speaker, Dr. Amit Asthana sir, who has spared his valuable time in his busy schedule in his research. As I have said, he is working more than 20 years of experience, and he is working with different kind of innovations, all these things, research and all these things. So thank you for that, sir, for spending valuable time with us, sir. So next is not the least, our participants who are always continuing with us from the starting of our webinar series and listening our talk from in this pandemic. And I hope all the people should be in safe conditions. So next, almost I have to thank our God, because of whom we are safe, and we are very much thankful for that. And with every time I used to conclude the session with some your quotations, or with some ethics. That is, today's ethics is your success is not final, and the failure is not the fatal. So it is the courage to continue that counts. So with this, I like to end today's session, and I like to hand over to our coordinator, Dr. S. Balamurali, to give the information for the next webinar series. Please.
Yes, sir. Thank you, Sahu sir, and thank you, Amit sir, for your valuable time and enlightening our participants with your thesis, with your work. So thank you once again. Thank you so much. And dear
You're most
attendees,
welcome.
dear attendees, feedback link is has already sent to your emails. So please fill the feedback form to receive your e-certificate, and we'll update you about our next webinar in either WhatsApp or through the emails. Thank you for your patience and participation. Thank you, Amit sir.
Bye.
Thank you once again.